Hello, everyone. Thank you for joining LakePharma's webinar on reagents and assays for COVID-19 antibody response. My name is Walter Tian, Vice President, Commercial Operations here at LakePharma. I will be your moderator today. Again, thank you for attending. Before we turn over to Hua Tu, couple of housekeeping items. Number one, all attendees are on an automatic mute. But you will be able, and you're actually encouraged to submit your questions through the Q&A box on your toolbar during the webinar presentations. And also, this webinar is recorded for future posting. So let me introduce the speaker of today, Dr. Hua Tu. Hua is the founder and CEO of LakePharma. He started the company in the San Francisco Bay Area back in 2009. He received his academic training at State University of New York and Stony Brook, and also Cold Spring Harbor Laboratory. After starting his biotech career as a postdoc fellow at Tulare, he continued to work on drug development at Tulare and then Amgen. His ongoing aspiration is to help bring efficiency at scale to the life science industry. Before I turn to Hua again, just a reminder, you can submit your questions through the Q&A box on the bottom of your screen under the toolbar. So now, let me turn over this to Hua Tu.
Okay, thanks, Walter. All right, slide number 4. So what I would talk about today are the LakePharma reagents and assays related to COVID-19 antibody response, and specifically will be related to the serological assays. Let's go to the next slide, slide number 5. So a quick word about LakePharma. Well, just to be clear, LakePharma is not a diagnostic ELISA kit manufacturer, and LakePharma is also not a clinical testing company, but instead LakePharma is a US-based biologics CRDMO. That stands for Contract Research Development Manufacturing Organization. The company has operations in San Francisco Bay Area, two sites outside of Boston and a new site in Dallas, Texas, and presenting this live from Dallas, Texas. LakePharma is a biologic company, and the company makes proteins and antibodies, as well as a range of other biologic products. The company provides integrated solutions on research, development, as well as GMP manufacturing. In this presentation, the key points I want to make is that LakePharma has been developing and testing various reagents related to SARS-CoV-2. And the reagents may be useful for serological assays and antibody development. I will show some data. And I also want to tell you that we're making the reagents available to the community. As an introduction for serological assays, serological assays against this virus has been in the news a lot recently. And as a brief introduction, serological assays are meant to detect the presence of antigen-specific IgG and IgM in the blood samples. And the various formats that have been developed and they're fairly standardized, they include ELISA, point of care, as well as multiplexing such as MSD. In the diagram on the upper portion of the slide illustrates how serological works in general. This is one example where the antigen is linked to a solid surface, and if there's IgG or IgM or other immune molecules in the sample, the IgGs will bind to the antigen, and then there'll be a secondary antibody, a detecting antibody against IgG or IgM added to the solution. If there's presence of IgG, then the detecting antibody will be co-localized with the antigen, giving a signal, and in the absence of an IgG, then there would not be a signal. So this can be used as a a binary positive or negative readout, or if this is done right, this can also be quantitative readout. In this kind of assays, the anti-IgG detecting antibodies, those are fairly well established. They're not specific for the antigen. So the key part is really the antigen. And we believe it's critical that our medical community not only get this kind of assays fast, but also get this right. And the quality of the antigen is the most important thing in the serological assay development. With regard to this COVID-19 pandemic, the most important protein on the SARS-CoV-2 virus is the CoV-2 spike protein. What we show here are pictures we took from the RAP paper published by UT Austin last month and published in Science. The diagram on the left shows the structure of the spike protein, and the spike protein forms a trimer. Okay? Like most viral surface proteins, they form a trimer. There's extensive interaction among the various subunits. And it forms a trimer, and the protein also has the transmembrane domain anchored to the virus membrane. So for us to make the protein, we will only make the active domain. So that includes everything from 1 through amino acid 1208 before the transmembrane domain. So there's no transmembrane domain in the full-length active domain. This S protein or spike protein has multiple conformations, and as illustrated in the RAP paper, in the Science paper published last month, they can observe by cryo-EM the up and down, let's just say up and down conformation, and with the receptor binding domain, which is colored in green here, and the receptor binding domain can bind to the ACE2 protein on the cell surface, and ACE2 serve as a receptor for the cell entry by the virus. So that's the general background about the spike protein, and it's the spike protein we're focusing on today. The reason we focus on this is because this is the protein, this is the most dominant protein on the surface of SARS-CoV-2, and it's the most natural choice as a target for the serological assays. So making full-length SARS-CoV-2 spike protein, that is something a number of labs have tried and have done, and we summarize here in that making this full-length spike protein actually is pretty challenging, and here are the reasons. One is that this is a pretty large protein, about 1,200 amino acid, so molecular weight is around 200 kD. And the protein expressed as a trimer. So the native protein is trimeric. So 200 kD times 3, we're looking at a very large protein complex, about 600 kilodalton. The protein is very highly glycosylated. There's at least a 20 glycosylation site on the spike protein alone. And we know that the protein has a furin cleavage site, so undergoes proteolytic cleavage. In addition, the full-length protein is able to adapt to different conformation, and this seems to be fairly typical for virus surface proteins, and it might be a mechanism how the virus can evade the immune system. This protein also seems to have propensity to be unstable, so far it's been a pretty difficult protein to make. Based on the reported data, this protein expressed at about 0.5 to 2 milligram per liter in mammalian transient production system. LakePharma has spent quite some time engineering this protein to improve the yield and also make sure we're making the right protein. And I want to show you some of the data that's on slide number 9. This is to make, again, the full-length active domain of SARS-CoV-2 spike protein. And we did this in a variety of systems, but the data we show here is in the TunaCho transient production platform. TunaCho platform is a LakePharma proprietary CHO transient platform. The parental cell with TunaCho is CHO K1, and which is the most common CHO used in the biotech industry. We showed 3 constructs here. We made a number of constructs, but this 3 constructs illustrate the protein pretty well. If we start with the graph in the middle, which is the SDS page of the spike protein in the pre-fusion conformation with the furin site intact, that is, we leave the furin site there, what we see is that we can observe the presence or the production of the full-length S protein labeled as S, but then we see 2 species, and we later realized one is the top one is S1 and the bottom one is S2. Based on the molecular weight, the protein, the full-length protein is 200 kilodalton, and the theoretical molecular weight of the protein should be about 130 kilodalton. The added molecular weight is really all due to the glycosylation of the spike protein. With the furin cleavage site, S protein is cleaving to S1, which runs at 120 kilodalton on our gels, and S2 running at 80 kilodalton. When we engineer out the furin cleavage site, we can see that there's no more S1, S2 formation, there's no more cleavage. Instead, we're seeing an intact 200 kilodalton protein. We see that with the wild type S protein with the furin site removed. That's on the SDS page on the left side. Pretty pure protein after 1-step IMAC purification, so 200 kilodalton. And as the gel on the right shows, that's the pre-fusion conformation with the furin site removed. So that's also, that's the protein, 200 kilodalton. So we're able to make the COVID-2 spike protein in the tuna CHO system. In fact, tuna CHO produces this fairly well. And the productivity we're getting is in the range of 30 milligram per liter in the 7-day production. We took the purified protein on SEC, looking at size exclusion, and we can detect that the purified protein runs about 700 kilodalton. Now, it's a pretty broad band on the broad peak on the size exclusion. And we're not seeing the monomer formation. In fact, the only other peak we see it is an 8 kilodalton molecule, which that's not the spike protein. And that's a protein we can easily remove by secondary purification. So the conclusion we see here is that the full-length ectodomain of SARS-CoV-2 spike protein is trimeric. It's fairly clear. We ran a number of molecular weight standard. That's what we see. And our data is pretty consistent with what the UT Austin group showed in their science paper. They showed it around about 670 kilodalton, so it's fairly comparable. So that's the full-length spike protein. We also made others, and what I show here on slide number 11 is the transient production of the RBD domain of the spike protein. In this case, it's an N-terminal histag version of the spike RBD. In the 7-day transient production in tuna CHO, we can see pretty good expression, 1-step purification, as shown on the CESDS here. They're pretty pure, single peak, and the molecular weight here is a bit higher than the theoretical molecular weight, which is 30 kilodalton. Here we're seeing higher, again, it's because of the glycosylation on the RBD. The next slide is we made the same protein, but this time the version is RBD with a histag at the C-terminus. We're seeing similar result. Again, pretty good expression, 1-step purification. The titer looks pretty pure. Then we took the protein to ACE2 binding. We want to test the interaction between the spike proteins we purified and the human ACE2 Fc that we produced in a variety of assay format. On this slide shows the ELISA data. Let's start from the top. With the spike RBD, we can see fairly good binding with both versions of the spike RBD to ACE2 Fc. And when we look at the full length S protein, we're seeing even stronger binding. It's about 10-fold to 100-fold more potent using the full-length protein compared to RBD. So they all bind, they bind very clearly. In this case, we coated the ELISA plate with ACE2Fc. We added the protein solution, we detected with an antibody that's specific against the his tag. We can see the binding pretty well. Interestingly, if you look at the different versions of the spike protein, in this experiment, of course, we need to do a lot more studies. In this experiment, we can see that the S protein in the pre-fusion conformation binds more tightly to ACE2Fc compared to the wild type spike protein. So there may be a mechanism for that, but we need to do more study on that. So ELISA shows pretty clear binding, so that's good. We also took those proteins to octet, looking at the interaction in the octet format. Again, having ACE2Fc protein on chip and then flow various spike protein we made. Here shows the RBD. Again, the two versions of RBD was seen binding with the apparent KD at about 16 to 17 nanomolar. So there's interaction between RBD and ACE2, although the interaction is, well, it's actually a very reasonable number. Once we put in the full-length spike protein, we saw something interesting here. So the full-length spike protein actually bind very tightly to the ACE2 on octet, and very strong signal. That kind of binding curve is comparable to antigen and antibody interaction that we normally observe on octet. So anyway, so something interesting here, it appears that there's a very tight interaction that we could observe between ACE2Fc and the trimeric spike protein. Now, there's probably quite a bit of avidity effect here, because the ACE2Fc is a dimer and the spike protein is a trimer. So there's actually a lot of opportunity for that interaction to happen. So I think what's interesting here is perhaps one can take advantage of the tight interaction between the virus and the ACE2, perhaps as a way of either detect or purify, or perhaps even neutralize the virus via this interaction. So anyway, we're pretty sure about the binding, that in fact the tie binding between the spike protein and ACE2Fc. So, so now we have those proteins available, and we're currently in production with some of the protein, and we've been increasing the lot size and as well as building a manufacturing process. In fact, we started building a stable cell line based manufacturing process about 3 weeks ago. So that as the stable cell line comes up, and we'll be able to use them as a production. In the meantime, we continue with the transient production to make those proteins. We're producing at this point several lots a week, so that gives us an opportunity to characterize as well as minimize the lot-to-lot variation. In addition, we're in the process of establishing release assays for those proteins for in vitro diagnostic application. And the release assay we are planning to establish is that we want to see greater than 90% purity by CE-SDS or SDS page, and greater than 80% trimer form by SEC, HPLC, or UPLC. And we definitely want to utilize the binding of ACE2 as a release criteria for the spike protein, whether it's the full length or the RBD domain. Because those proteins are used for in vitro diagnostic applications, we have no requirement for endotoxin or bioburden testing, so those will not be included. In addition to building the release assays, we're also working on the formulation and stability of such protein. As I mentioned earlier, some of the proteins are not inherently stable. They're pretty difficult to make, and we have a desired product stability profile we want to establish, and we want to see at least 14-day stability at 4 degrees storage, and we want to see long-term storage at -80s, and also the samples can be shipped on dry ice. So you can imagine we're doing a number of freeze and thaw and stress testing of the samples we produced in order to establish the stability profile. All right, so some of those proteins available, and on this slide, I want to tell you how you can get the proteins from LakePharma. We have generated a material transfer term sheet for the COVID-19 spike proteins and related reagents. There's a file already available. And again, I want to specify that the proteins we provide are for in vitro diagnostic assays as well as antibody testing only. Those proteins are not not suitable for injection into humans, not as a drug substance and drug product. In fact, the current formulation of our protein, because they're for the in vitro diagnostic purposes, we may actually include azide as a preservative in those proteins. So again, they are not for injections in humans. And the recipients of the material cannot distribute or resell the products to another party. In terms of availability, we have full-length spike proteins available. We also have the RBD proteins available. The ACE2FC protein is also available, although we're only giving those out in small quantity because they would only be used as a testing or a validation for the spike protein. Currently, there's limited availability because we only started producing this in various lots. We only started that about 3-4 weeks ago. So we're still ramping up the production. So if customer wanting to get those proteins, please contact us and make request. We do not have this product sitting in refrigerator ready to ship. So those do need to be requested in advance. And right now, whatever amount we produce, they get sent out almost as soon as they get QC released. So every protein will go out, and we have the lot number system, and we do encourage the customers use and reference the lot number in communication with LakePharma. And we will in turn share that results with others. So on this slide number 17 shows our current price. We develop a price sheet for nonprofit medical center and universities first, because we've been working with several nonprofit medical centers for the past few weeks. So we established that we want to make the material and we want to make it priced as good as we can. So we price those material at cost. The full-length spike protein, the trimer form with the C-terminal histag producing the tuna cho, we are making those available to medical centers at $500 per milligram. So that's our cost. And if you run the economics here, if you're using 0.1 microgram per well in an ELISA test, that represents about 5 cents for the cost of material per test. And if you run it at 0.5 microgram per strip in the lateral flow test, that represents 25 cents per test. With RBD, because the protein produces better, we price that lower for medical centers. And universities, we price it at 50 milligram per liter. So then that's where you start to see your ELISA test, the cost of material will be even lower. In addition to research grade material, we are planning to do GMP grade should there be interest in doing that. LakePharma Hayward is capable of GMP manufacturing for ASRs. And so we're ISO 9001 certified. And so those are work in progress, and we're going to be in pretty good scale fairly soon. Those are the current price. Please know that the price may change, and as we increase the scale, and as well as change the release criteria or make so that could change. There's quite a bit of information and resources on LakePharma.com. We have a dedicated page on the website that lists all the relevant reagents that we have produced. That's LakePharma.com/integrated solutions /COVID-19-activities and COVID-19 key reagents. So there's a number of constructs there, a number of material there. I can tell you we are in the process of making spike proteins from other known coronavirus. That includes SARS, MERS, and the two beta coronavirus that are putatively in our seasonal flu lineup already. So those material can be used as controls. At the same time, we are also making recombinant IgG and IgM in the human form against the spike protein, and they have the potential to be used as references and standards in the serological assays. And if you want to connect with LakePharma, we'll certainly welcome that. And if you're an existing client of LakePharma, the fastest way is contact your account manager. If you're new to LakePharma, you can establish the connection by emailing us at kick-covid@lakepharma.com, or you can use the contact us form on the website. And we do welcome feedbacks, comments, and suggestions. lakepharma.com/feedback, and that's where you can feel free to reach out to us. I'm near the end of my presentation, so we do like to collaborate with various parties. So, and I want to show you a few areas that we have interest. One is that LakePharma would like to validate those protein products I just mentioned. We like to validate that with serum samples before distribution. This could save the users quite a bit of time so that they don't have to do the validation individually in their own lab. So we would welcome to receive human samples that are antibody positive and virus negative. We cannot work with virus positive samples in LakePharma. That's not our, we would avoid that kind of samples. We're also interested in collaborate with folks to quantify the serological test, so quantify the immune response. And probably more interesting is also we would like to develop reagents and assays that can measure not just the presence of antibody, but the presence of neutralizing antibody. So we have significant interest in that domain, so we would welcome collaboration. A bit outside of the serological test, I want to mention that LakePharma we have a lot of interest in the protein vaccine space for this virus. And LakePharma has 2 GMP facilities that can be used to support this kind of diagnostic reagents as well as actual drug substance manufacturing. Our ASR manufacturing is in Hayward, California, and our mammalian drug product manufacturing is in Hopkinton, Massachusetts. Again, to reach out to us, please email us at kick-covid@lakepharma.com. Your inquiries will be answered by the customer support team based in Dallas, Texas. Okay, so this is my last slide, and really like to thank everyone who works on COVID-19 and work on this important problem. I think this really takes on a community effort to solve this, and we're glad that we're part of that community, and we want to be helpful as much as we can. I'm also appreciative of LakePharma teams who have worked hard through this pandemic, and I think our materials and our assays would be useful for folks. And I also like to thank you for your attention, for joining us at this webinar. Okay, thank you, Walter. I'll turn that over to you to see if there's any questions.
Thank you, Hua. Thank you for that inspiring presentation. We're really proud that LakePharma is making real impact in this global effort fighting coronavirus epidemic. So now we move into our Q&A session. Hua, can you transfer the control back to me?
Yeah, so one question is, what's the meaning of pre-fusion confirmation of this? So I would say the pre-fusion confirmation is probably the most interesting thing that came out from the virus research in the past 10 years, is that to recognize that viruses, the surface protein, actually have different confirmation. The pre-fusion confirmation is referring to a particular confirmation where the virus is ready to attack, ready to attach to cell surface, and ready to go through a cell entry process. There's a lot of literature on that, and at this point, taking a virus and make that into a pre-fusion confirmation is fairly well established. So there's a lot of literature in the public domain. The second question is, do furin cleave generated protein co-migrate in SEC? That's interesting. I can't say for sure on the SEC, but I can tell you that we mentioned this on there's a page on the LakePharma.com. We actually mentioned this. After cleavage, we believe the S1 and S2 are still associate. Okay. Maybe that's what the question is about, is whether do they associate. I think the answer is yes, they do. They have the potential to fall apart, but a lot of it is still attached together. So, third question is, the transient production plasma available for nonprofit? The answer is sorry, no, it's not available. It's in the LakePharma proprietary vector. We cannot distribute the DNA. Fourth question, how about the cost of private companies? Yeah, we have developed a price sheet. It's a bit higher. It's a little bit higher than the nonprofit. That information is available. You need to speak with either your account manager at LakePharma or contact the customer support at LakePharma. The next question is, does RBD N-terminal histag bind to S protein better? Is the octet binding data, the binding curve for this protein, shift a bit to the left? I actually don't understand the question. Maybe the question needs to be refined. The RBD does not bind to S protein. We didn't check for that binding. In both cases, we were looking at the RBD or the spike protein binding to the ACE2 FC. So our data is very clear. The full length S protein bind to ACE2 much tighter than the RBD. So the interaction between RBD and ACE2 is about 16, 17 nanomolar apparent KD, whereas the trimer binds much tighter than that. We believe it's because of avidity effect. So, but what's interesting here is physiologically there's already avidity, so that avidity is real. It's not an artifact. The next question is, do you have instruction protocol of using spike protein to create ELISA plates? Well, that Mount Sinai has published a protocol on ELISA using the full-length spike protein as well as RBD. So that is already in the literature. So next question, does use of RBD protein generate a lower level of cross-reactivity in serological as when compared to full-length? Yeah, I think the answer is yes. Because RBD is a smaller protein compared to the full-length spike protein. So now which one's better? We don't know. I think there are pros and cons of using full-length S protein versus the RBD. I think with RBD is probably more focused on neutralization or the interaction with ACE2, because ACE2 is only binding to the RBD domain of the full-length spike protein. We're not aware of another interaction, another domain on the spike protein other than RBD that binds to ACE2. So I think the answer is yes. Use RBD will have a lower cross reactivity compared to full-length spike protein. Is the slide deck available on the website? I think it will, but I think the video will be available on the website at some point. Video of this webinar. So in terms of availability, small quantity. So if you're looking at milligram scale, if we can't ship today, we can ship tomorrow. Okay, it's pretty available. What we cannot ship right away is if you order gram scale of the protein. That will take us maybe 2 to 3 weeks to get to. So, and do we ship to Europe? I think I don't see why not, but at the moment, we do prioritize, we do give high priority to medical centers and universities and the government agencies over diagnostic companies. So I think that's probably a bigger focus on in terms of priority. And I think there's one question that's interesting. The question is, would the full-length spike protein need to be handled in a biosafety laboratory? And does it require BSL-2, 3, or what? The answer is what we provide are recombinant proteins, and they're not viruses. They're not a portion of a virus. They're recombinant proteins producing the mammalian production platform. So you actually do not need to treat it as a virus product where you need to consider for the BSL-2, 3, or so. Yeah. Okay, so I think that's I think that's it for the Q&A.
Thank
Back
you,
to you,
Hua.
Walter.
Again, thank you to all attendees for joining us for this webinar. We will, number one, make sure we follow up on all the questions that are raised during the webinar, follow up on one-on-one. But also number two, just as a reminder, the recorded webinar will be posted on LakePharma website. We will let you know as soon it is available. Again, thanks everyone, and have a great day. Take care.